Finally, the last video, the signal transmission at synapses. Yay! So we got a couple of different types of synapses going on here. We got an electrical synapse, which is kind of nice. It's pretty much just the neuron attaching directly to the next neuron and sending those sodium signals or calcium signals straight on through, and it's really quick. It's wonderful. It's through a gap junction. But the majority of your synapses is a chemical synapse. Chemical synapse, meaning that it's just the terminal end, the axon terminal is just barely hanging just any bit above the top of that here neuron. But it's not actually touching. It's kind of like that little brother goes, I'm not touching you, I'm not touching you, I'm not touching you, I'm not touching you. Giggles type of thing. But in between, there's still, you feel that sweaty weirdness that is your little brother's hands. Those, I guess you could compare it to that of the signaling molecules. Now, really quick before I go on to the next one, there are 3 different ways that a neuron or axon terminal of a neuron can interact with the next one. It can go through the dendritic, axodendritic axon. Dendritic, it's saying that this axon is associated with the dendrites. This is probably the majority of the things that are going on right here, and it's because it has a lot of those receptors. It has a lot of those things hanging out right here. Axosomatic, somatic means body because it is interacting with the neural body itself, while the axonic actually goes ahead and interacts with the very beginning of the axon at the axon hillock, which if there's even a change in that gradient potential right down to negative 55 millivolts, yes, you are going to have it happen. Now, as I was saying about the little brother saying, I'm not touching you, I'm not touching you, I'm not touching you. Yeah, you're right. So, but it's just ever so close that you can feel some type of interaction. So what's going on is that pulse, that sodium pulse, that action potential is getting down to these terminal buttons right down here, these synapses. And what it does is it causes calcium to come in to the channels. And this calcium causes the release of these vessels, these synaptic vessels. These synaptic vessels are just packed full of neurotransmitters, whether it be a chemical or a gas or some type of compound, what have you. It just releases it back out into the synapse, where it will interact with ligand-gated channels. They interact with just enough, they can do a myriad of things. And for instance, one thing they can do is have a positive thing where they let in the sodium channel. Now, really quick, just a little bit of terminology. Synaptic cleft is that space between your little brother's finger and your face or what have you. Or in this case, the axon terminal with that of the dendrite, the neural body, or your axon hillock, forgive me. Now, you may have noticed that there isn't really, those things could possibly drift out. That's where your astrocytes and your microglial cells come in and kick in and grab those up. And also, if you want to recycle them, they can be pulled back up into the axon terminal, or it can just be destroyed on the outside. Either way, it creates the type of impulse that it may or may not want. Now, there's a couple of ways that this ligand will interact. There is an ionotropic receptor or a metabotropic receptor. Ionotropic is just the ligand interacts directly with the channel and opens it. It's these two right here, whether it be excitatory or inhibitory. The metabotropic one is where the ligand comes along, binds into some type of protein, which causes a cascade of events, one of which will be opening up a channel. This is for instance of an inhibitory postsynaptic potential. Now, as I had mentioned a little bit, the excitatory inhibitory goodness. Excitatory is just depolarizing. If you go back to the second part of this little chapter 12 series, you'll remember that the depolarizing, it gets closer to that 0, while the inhibitory does a hyperpolarizing, you're getting away from that 0. Now, you will see multiple neurons interacting with a single neuron, each of which trying to increase or decrease that stimulus. If it's an increasing of that stimulus, it's going to be an excitatory postsynaptic potential. While if they're trying to inhibit it, it's going to be an inhibitory postsynaptic potential. Now, to help you remember just a little tiny bit, going back to the muscles, the tetanus, we're going to go ahead and just revisit it. What it does is that tetanus toxin or that the bacterium will go ahead, sneak up, and block off the inhibitory neurons. If it blocks it, that means, hey, free reign. That hyperpolarizing events, the things that make certain your muscles are nice, smooth, calm, relaxed, and aren't fighting against each other are now gone, which means even the slightest excitatory pulse, if it gets it up to that action potential, is going to trigger. Hence why you get lockjaw or your muscles are tense and all that wonderfulness that is tetanus. So, real quick summary, remember dendrites. Dendrites is where the majority of your other neurons are going to interact. The cell body containing all the necessary things to survive, followed by the axon. Usually there's only one axon hillock which comes off. It may branch off, but most likely it won't until the axon terminal. Now, let's see here. I wonder if that actually recorded. Oh well, if it did, it did. Happy birthday. You've just become famous. If a summation is when all those signals coming from those different neurons are all coming together via is called a spatial summation, meaning that you got multiple neurons coming in trying to shoot up and hit up that negative 55 millivolts up to that threshold and cause something to happen. A temporal summation is just, remember, temporal means time. So if over time this presynaptic neuron is shooting as much as it can eventually trigger that nerve impulse or that action potential. In epilepsy, there's just that uncontrolled firing of those synapses that it just depends on how severe and what places it happens. It's just out of control. So just what I meant by depending on the places, if it's a focal seizure, usually it's a localized location, whether it be an arm or you're smelling something that's not actually there. Usually you record, it doesn't really have to be with a loss of consciousness. While more of the generalized seizure is the stuff, this guy right here is the stuff that you're more aware of and used to or think of. There's 6 different types, all of which referring to some type of loss of muscle control and possible consciousness. The one that everyone thinks of when we think seizure, though, is the tonic-clonic, which used to be called the grand mal seizures. It's much more dramatic. The person flops down to the ground, is pulsating, you can't control himself, all that wonderfulness. I should say wonderfulness, that's definitely not fun. With that, let's go ahead and talk about the different types of neurotransmitters. You got quite the group of different types of neurotransmitters going on here. Everyone's familiar with acetylcholine. There's that it is in all of its glorious beauty, skeletal beauty of the chemistry that is life. You also have a lot of different types of things that could be secreted out of a axon terminal into a synaptic cleft, such as amino acids or nitric oxide, carbon monoxide, surprisingly. The biogenic amines, that's pretty much just referring to a aromatic molecule at the end of a chain with a variety of different things, as well as purines, the things that you can, like adenosine triphosphate. Purines referring to, if you remember back to good old DNA, RNA, this guy right here, adenine, has a role in building up that skeleton. And then you have your neuropeptides, so things that are completely made up of a chain of proteins. Now, we could go into a lot of detail about all of these, but their effects are wide and different. Why is that? Because there's different types of receptors going on. So for instance, a norepinephrine and epinephrine usually have some type of role in vasodilation at certain points, but can have vasoconstriction, or they They either open up a blood vessel or clamp it a little bit tighter, depending upon where they are. Dopamine, serotonin have a pleasing, happy effect unless in the wrong spot. Acetylcholine could be a, a positive thing or it could be an inhibitory thing. Here is the list of neuropeptides that you will see again. These two you will definitely see again, angiotensin 2, cholecystokinin. Don't worry about that. Um, the ones that I do care about a little bit are probably substance P and enkephalines, endorphins, and dynorphins. Go ahead and check those out. Learn a little bit more about them. But for now, we're going to go ahead and just move on to the neural circuits. So there's a different types of neural circuits. We're going to look at 5 of them. I believe I actually have a picture of just 3 of them. We'll take a look-see real quick. Here, let's look at diverging versus converging. A simple circuit first is just one after another. It would be like what this neuron is attached to this neuron, one after another. A diverging circuit is you got one that might interact with 2, and those 2 interact with another 2, and it just propagates the thing. A converging thing is everything's coming together. A reverberating circuit means that this guy right here can actually go back and interact with other neurons, which will reactivate it over and over and over and over again. Parallel circuits, that's referring to a neuron that interacts with 3 different neurons, but they all eventually come back down together onto one specific output. Finally, I believe this is the last topic we're going to be talking about. How we take care of the nervous system if it gets damaged, as well as how it adapts. Plasticity refers to its ability to move and change and relocate. And regenerate means to be able to repair or heal. Neurogenesis, it begins nice and pretty, you build up your brain, but afterwards it's pretty much, yeah, you're not regrowing at all. There's little or no repair. So, which is a good thing. You don't really have much space in your cranial cavity, and if you let your neurons grow and grow, you would run out of space. But it sort of has that downfall where it doesn't let it grow back. Those of you that want to become a scientist, maybe this is your chance to figure out how to overcome that issue or those inhibitors, that is the oligodendrocytes. So here's a really wordy thing. Here's a less wordy thing. I like pictures. So here's a normal cell. Oh no, it got damaged. How sad. So the Schwann cells, when it notices it gets damaged, will actually form this Wallerian tube or this Wallerian regeneration. And it will try and regenerate. It'll create a tube where it can be regenerated. Axons can be healed, but if you get a cell body, not so much. With that, that is it. If you have any questions, feel free to enjoy yourselves to watch it again or send me an email, send me any type of communication. Hope you have a pleasant day.